Hello, welcome to part 2, the remaining sections of chapter 2. I'm going to start my timer, and while I'm doing that, put your phone off or on mute and get yourself some space and some time, about 30 to 60 minutes. This, I know I went through the first part, which was the the chemistry review, and I realized that after almost every single slide, I said, oh, and do you remember this from chemistry? So I promise I'm going to try to stop doing that, except for one. So this rest of this chapter, you should remember from biology, because it talks about water and its properties and how that's important to life, a little bit about pH, and then it goes over the 4 major organic compounds. So this should be stuff that is all a review to you, although it might have been a while since you've had biology. We're going to start with water. So if you remember, the chemical composition of water is an oxygen with 2 hydrogens attached. If you would flip this over, it almost looks like a Mickey Mouse head with ears. And the hydrogens form a hydrogen and the oxygen form a hydrogen bond, whereas covalent bonds, which are the shared electrons, are formed between the 2 hydrogens, oxygens and the 1 oxygen of the water molecule. Water's properties, you probably went in biology talking about many more of these. Oh, I promised I wasn't going to do that. You might know more of these properties, but these are the ones that are physiologically important. And then the next section talks about a little bit more about that physiology and the systems that depend on it. First of all, it's the most important component of your body. It makes up a lot of important systems. It's a big component of a lot of important systems, and it's about 2/3 of your total body weight. And it touches, even if it's not a direct component of the system, it affects all the systems. The first property is lubrication. Lubrication is important because your joints, anything with friction. So think about, we just talked about in the last chapter, all the membranes, all the serous membranes. Those serous membranes in between the parietal and the visceral have a liquid, and that liquid is mostly water, and it's an effective lubricant. All of your joints also have these pockets where there's lubricant, and it's mostly water. So a little thin layer will reduce some friction between these surfaces, and that's very important for human bodies. The other properties, this is on page 69 in your textbook. Another important property that will kind of move us into the next system is the idea is that it's a solvent. A solvent is something that does the dissolving, and so water is a very, very important solvent. And I think we talk about solvency right here. The solvent and solute, the solution is both the solute and the solvent. But let me go back because I accidentally skipped. Heat capacity is also very important. It has a relatively high heat capacity if you compare it to other chemicals. And this means it takes a lot of energy to actually break the water molecule, or the bonds between the hydrogen bonds between the water molecules. And so that's helpful because think about on a hot day when you sweat, the liquid from your sweat is actually evaporating from your body, and that evaporation is removing some of the energy. It's actually reducing your body heat. It's a way of homeostasis, if you remember from chapter 1, maintaining that steady state. Okay, sorry about that, jumping around in that section. But the next section is about physiological systems and how they depend on water. So many of these inorganic compounds are held together by ionic bonds, not covalent like water. But water is able to break the ionic bonds, and that is called ionization or dissociation. And it's because of its structure that's able to break these. It's because it has the slightly positive end and then a slightly negative end that's actually able to break up these inorganic ionic compounds. So in an aqueous solution, anions are surrounded by positive poles of water, and cations are surrounded by negative poles of water. Okay, so the negative side. A pole is like the north and south pole. It's not really up or down, left or right, but it helps to describe a sphere. Oops, sorry. And this forms these hydrogen spheres, which it has a layer of water around these ions in solution. So then molecules with these polar covalent bonds, just like water, also attract water molecules, but they are attracted in a different way. The water forms hydrogen sphere around the molecule, and if the molecule binds to the water strongly, it'll dissolve, okay? This is a hydrophilic situation that is water loving. Hydrophilic, if, you may have heard this term before, means that the substance will dissolve in water. It'll dissociate in water. Glucose is an example. Aqueous solutions with anions and cations also conduct an electrical current. And this is important when we start talking about some of these physiological systems that actually depend on a difference, a gradient in these ions. Electrolytes are these inorganic substances that will dissolve in water, and the ions will conduct an electrical current. There are a lot of salts, not just sodium chloride, like calcium chloride. These are electrolytes. In an electrical field, the cations, because they are positive, will move towards the negative side, and the anions will move towards the positive side. In this picture, along with a good graphic here of all the stuff we've talked about, is on page 70. And continues on page 71. Again, so why electrolytes are important is because we need this for systems like the nervous system and the muscular system. But there's also concentrations of ions that need to be carefully regulated, carefully contained, part of homeostasis. And that includes organs like the kidneys, which help to control what ions are excreted, how much water is excreted. Digestive tract also absorbs a lot of ions and water. The skeletal system is a great storage system for ions like calcium. We actually have cells in the skeletal system that are very specialized to either break down and release, or to build up that calcium. And so here's some important electrolytes that are important to us physiologically. So the next page is about the hydrophobic, the importance of hydrophobic molecules and compounds. Hydrophobic, water hating or water fearing, means it does not dissolve or is soluble in water. And so that's important. Fats and oils are very important hydrophobic substance. And, you know, we have lots of fats and oils in our system. Think about your hair. Your hair follicles have a little a sebaceous gland that secretes it's called sebum. It's oil, okay, and that helps to keep your hair flexible, okay. And these are all non-polar, which means there's not a lot of a charge. There's not a positive or negative pole. It's kind of the opposite of what, you know, the polar molecules are, obviously. And so these kind of sit. I mean, think about Italian dressing. Italian dressing has an oil and a vinegar base. You can see the oil, if you let that Italian dressing just sit, you can see the oil float to the top. That's because it's hydrophobic. Large organic molecules held in solution by these water molecules are called colloids, all right? Think about, and the example they give you is liquid Jell-O. It's a very thick colloid. These will remain in solution indefinitely, And usually when they, the solution contains large molecules. Suspension is a solution containing even larger molecules. For example, whole blood is a suspension of plasma, is the solution, the matrix, and then it has red blood cells, all different kinds of white blood cells, and little platelets. All right, now we get into the pH, and if you remember the pH scale, which is on page 72, and it might come up right here, the lower the number, the more acidic the substance, okay? And if it's more acidic, that means it has more hydrogen ions. The hydrogen loses an electron, becomes positively charged, and that becomes extremely reactive. So a lot of the acids that you may have heard of have like HCl or H2SO4. You know, those, they start with that hydrogen in their chemical formula. The bases have less H pluses, but they have more OH minuses, which are the hydroxide ion. And water, as you know, is a combination of H's and OH's. So the more OH's it has, the more basic it is, or the higher the number. And that just, I'm repeating that right here. Neutral is going to be equal to about 7, where the H's and OH's are about the same. And here is that picture on page 72 that shows you the entire spectrum here of going from very acidic to very basic. And here's some things like saliva. You know, you notice that's not exactly neutral. Blood is a little bit basic. You know, urine can be, there's a range depending on, you know, the amount of water that's released. So those are, you know, 3 substances in the body that it's important that they keep that pH. Anything below 7.35 is called acidosis, okay? And it's, and pH below 7 can cause some serious problems like coma. And anything above 7.45 is called alkalosis. It means it's basic, and your skeletal muscles will not operate properly at that pH. This is an example of an acid here. These are just some important substances coming up here that are important to the human body. And again, it's a review. Acid, like hydrochloric acid, it means it's losing an H+. They're also known as proton donors. And there's a strong association with some of these stronger acids. A base is something that is higher on the scale. It releases an OH. Sodium hydroxide is an important example. Weak acids and bases will not dissociate completely. You see this reverse reaction here. They will actually kind of slide back and forth. And carbonic acid is important because carbon dioxide is something that's in our blood, because that's what our cells use, or that's what our cells make as a waste product, and then we exhale some of it. So it reversibly dissociates into hydrogen ion and bicarbonate ion, and the acid comes from, you know, our breathing in and out, and our cells metabolizing. Salts, these can be important electrolytes, or they can be important ions for things like muscle contractions. So these are also very important. Buffers are things that help to stabilize or maintain the pH of a solution, and usually it does that by either dropping off or taking up hydrogen ions, and then it kind of helps maintain that pH. Usually involves a weak acid and its related salt. For example, carbonic acid and sodium bicarbonate. All right, so you might take a pause here on page 74. There's a good review of all the ideas about water. So take a pause and see if you can't answer some of those questions on page 74. The last section here that we're going to talk about is about the organic compounds. There are 4 main organic compounds: carbohydrates, proteins, lipids, and nucleic acids. The book goes through a little bit of the chemistry of those and then its importance in the human body. So we're just going to review past all 4 of those. In general, organic compounds contain carbon and hydrogen. They have a functional group attached to them, which makes them all different. There's some common functional groups, which will be in a second here. There's a table on page 75 that shows you some common functional groups. So, and here it comes. The amino group, things like amino acids have an amino group. That kind of makes sense. A carboxyl group is that COOH, which are in fatty acids and amino acids. Hydroxyl, that hydroxyl is a lot of different places. That hydroxyl is very important because that can be lost with another H in a dehydration synthesis, and you can build some larger molecules. And the phosphate group, nucleic acids, phospholipids, those are important examples. Okay, carbohydrates. Carbohydrates, as the name implies, carbon, carbohydrate, as in water, H-N-O. And it's usually in a 1 to 2 to 1 ratio because hydrate, water, is H2O. So that would be your 2 to 1 ratio of hydrogens to oxygens. And that ratio is usual, but not 100% all the time. It's about 1.5% of the total body weight, and it's a very important energy source. There are certain classes: monosaccharides, disaccharides, and polysaccharides. The monosaccharides and disaccharides are the more simple sugars, and the polysaccharides are more complex carbohydrates. In those complex carbohydrates, there's a lot of stored energy, a lot of potential energy that the body uses. Some more information: monosaccharides contains 3 to 7 carbons. This is a glucose molecule that you could see in the chain form or in the ring. Isomers. Isomers are molecules with the same molecular formula, but they're different structurally. Glucose and fructose are examples of that. And so you would have the same molecular formula, C6H12O6, but they're different. And you can see how they're different by the drawings on the bottom here. We talked about hydrolysis and dehydration synthesis in the last slideshow on chapter 2. But here you can see glucose and fructose. If it were to lose that HOH in a dehydration synthesis, which is going left to right, you would get sucrose. And when sucrose loses in HO and OH, you would, in the hydrolysis, which is right to left, then you would get glucose and fructose. It's important to note, just like we talked about enzymes in the last section, an enzyme, actually called sucrase, is going to catalyze both the dehydration synthesis, the left to right, and I hope my hands are going in the right direction, but the left to right dehydration synthesis and the right to left hydrolysis is both catalyzed by sucrase. Polysaccharides are those complex ones. Things like starches and glycogen are very important. Starches have they're a big energy source, and glycogen is also a big energy source. Starches are usually from your plants, and glycogen is usually from your animals. Polysaccharides usually have many branches, very large molecules in general. And here again, some examples of each of those, the energy source. I think this is actually repeated, that table on page 76. Next organic molecule is lipids. Lipids starts on page 78. Lipids are very important in terms of energy storage as well. Lipids contain carbon, hydrogen, and oxygen. Their carbon to hydrogen ratio is around 1 to 2. There's much less oxygen compared to carbohydrates, but there are similar numbers of carbon atoms. There may be smaller quantities of other elements like phosphorus, nitrogen, or sulfur. But the lipids are your fats, oils, and waxes. Those are mostly insoluble in water, meaning they are hydrophobic. And so they have a special place in transport mechanisms and in other areas like your earwax and the oils that are produced by your hair follicles. So here are some examples of lipids. Some other things like hormones and steroids are also lipids. Of course, cholesterol is a steroid that's a very important structural component of our cell membrane. It keeps our cell membrane nice and fluid so that things can very easily pass through it. Phospholipids are also a very important structural component of plasma membranes. And it's their makeup that allows things to pass. We're going to review all of that in the next chapter on cells. A fatty acid is a common lipid in a lot of foods that we eat. It has a carboxyl group on its head. The head is hydrophilic, meaning it's loving, and the tail is hydrophobic. This is similar to the phospholipid setup in that that duality there will allow it to attract certain molecules and then repel other molecules. Saturated fats. Saturated fats means that the carbon, if you look here, the carbon is saturated with hydrogen. So there's single bonds and there's a lot of hydrogens involved. Unsaturated means there's some double bonds so that this carbon isn't saturated with hydrogen. It's really only attached to one hydrogen. Saturated fats are more liquidy, and so they will kind of ball up and get thick. And then unsaturated fats are more, they're not more solid, but they're more fluid. They're not going to ball up like that. So your saturated fats, I mean, like think your butters and your lards, and your unsaturated fat, think of your vegetable oils. Glycerides. Glycerides are also very important. These triglycerides are one that you might have heard of. This is a glycerol and 3 fatty acids. And these are in foods that you eat. Also important energy sources. And here's how they're formed. You get this dehydration synthesis here. The fatty acid attaches to one of the parts of the glycerol. Some functions of lipids. They're essential components of the cells, as we said. They're important as energy reserves, and they provide twice as much energy as carbohydrates. So they're huge energy reserves. Some of these lipids are very important chemical messengers and structural components. So we're going to talk about some of the different types here in a second. The eicosanoids, it's a mouthful. These are lipids like leukotrienes and prostaglandins. And leukotrienes are produced by some cells in response to an injury. So they have a like an immune response, almost an immune response. Prostaglandins are also released by damaged tissues, and they can help with pain, receiving sensations of pain. Other examples, steroids. We said cholesterol. This is a really important one. There is good and bad cholesterol, and we're not going to get into a whole bunch of that, but basically the good cholesterol is the stuff that's in your cells, and the bad cholesterol is the stuff that, you know, balls up from having too much of that in your food, and that gets into your bloodstream, causes problems. Phospholipids, we said, was part of that cell structure. A glycolipid. Glycolipids can be also in the cell membrane as markers. In water, they can form these large droplets called micelles. That's actually really interesting because we've used these micelles as evidence that, you know, the cells can, how cells get together. Sorry about that. So we're still talking about lipids and the micelles being important. These large droplets, they actually gave the kind of precursor to when scientists were trying to discover how cells evolved in the first place. Okay, next are proteins. Proteins start on page 82. They are the most abundant organic molecules. They are about 20% of your total body weight, containing carbon, hydrogen, oxygen, and nitrogen. Most of them have sulfur and phosphorus as well. If you remember transcription and translation, they transcribed from the DNA and translated into amino acids. And so these proteins are these long chains of amino acids that have multiple layers that we'll get to in a second here. The amino acids are kind of all structurally the same. They've got the central carbon, they've got a hydrogen atom, an amino group, and a carboxyl group, and then R group. And there's only so many R groups that give us, I think, if I recall, 21 amino acids. Peptides are amino acids combined with or built together by dehydration synthesis. They are the covalent bond that forms between the amino acids is called a peptide bond. So a dipeptide is 2 amino acids and polypeptide is 3 or more. Peptides of over 100 amino acids are called proteins. And when I talked about, this just shows you the dehydration synthesis, but when I talked about the structure or the levels, the first level primary is the sequence. So whatever amino acid this is named, this is the sequence of the amino acid. Then you get into secondary structure, which is when you can either twist that primary structure, that sequence, you twist it, or you fold it. So here's a twist and here's a fold. The twist is called a helix, an alpha helix, and the fold is called a beta sheet. And those folds and twists form because of hydrogen bonds between usually the R groups. Tertiary structure is when you take those folds, all those folds along a big long amino acid chain, or you take those twists and you kind of ball it up. And so it gives us this three-dimensional shape, and that's called tertiary structure. Quaternary structure is resulting from multiple chains. So you take a tertiary structure and you add 3 more tertiary structures from 3 more proteins, and you get this hemoglobin, okay, which is a full functional protein. Proteins usually are not structurally, or they're structurally, but they're not functionally useful until you get to quaternary structure. Hemoglobin contains 4 polypeptide subunits. This is very important. It's in your blood and it attracts oxygen and carbon dioxide, so it can pick up oxygen in your lungs, transport it to all your cells, drop it off, pick up carbon dioxide and come back. Collagen is a fibrous protein that gives a lot of strength to tissues like your skin. And so this picture might help you to, this picture might help you to visualize that structure. It is on page 83. Denaturation is when there has been a chemical and structural change to the proteins, tertiary or quaternary structure. And so if you've actually changed that structure, then you've changed the shape and then you've changed the function, so it doesn't work anymore. This can happen in different pH ranges, different temperature ranges, different salt ranges. It just depends on the protein. But denaturation means you've killed that protein. And if that protein happens to be an enzyme, then you're in trouble. Enzymes are a very important protein. They have an active site, which is kind of like a parking lot, where the substrate, which is the reactant of an enzymatic reaction. The active site is where those substrates park. And then the enzyme, either breaks it apart or builds it. And this substrate enzyme fit is the lock and key theory. Here's kind of an overview of the process. You can see that these are two reactants, two substrates. They bind at the active site like a lock and key. The whole thing is called an ESC, or enzyme substrate complex. And then the product is released once those new bonds are formed. The enzyme is not used up. It actually can be recycled. Not an unlimited number of times, but certainly a lot of times. There's lots of enzymes. If we took out all our enzymes, we'd drop dead. I always say that to my biology students. Enzymes catalyze absolutely everything. And the fact that you can activate or inactivate enzymes is kind of a short-term control mechanism for a lot of these pathways. There's a saturation limit where you have kind of you've maximized your enzymes rate of reaction. The last one we're going to talk about are nucleic acids, and ATP is an important nucleic acid. This is a high energy compound. This is so when we talk about respiration, the cells are breaking down the large molecules that we eat into these small molecules. So it's like trying to go buy a pack of gum, but you only have a $100 bill. So $100 bill is like these large molecules. Well, your cells, specifically your mitochondria, are able to make change. So from that $100 bill, maybe 75 little $1 bills come out, okay? Those $1 bills are ATP, because our cells can use ATP from everything from transport, active transport, to contracting a muscle. There's some other forms of ATP. AMP is a monophosphate, ADP is the diphosphate, and then DP is the triphosphate. Each time you have a phosphate, you have a bond in between the last two phosphates. That's where the energy is found. And with ATP, that high energy bond is that last one, so that's the highest. And here it shows you this would be AMP, add another phosphate, this would be ADP, add another phosphate, this would be ATP. Okay, this is an example of how ADP and ATP are formed during cellular respiration. This ADP comes in contact with another phosphate because here's the energy, and it'll put it together to ATP, okay? And then when we use that energy, the phosphate is lost to be recycled and we lose energy. So this is a reversible reaction. Another view of that. Other nucleic acids are deoxyribonucleic acid and ribonucleic acid, DNA and RNA. We know these are very important in storing hereditary information. Nucleotide is the monomer, and it's composed of a phosphate group, a 5-carbon pentose or sugar, and then a nitrogen base. And the nitrogen bases, remember your A's, T's, G's, and C's, here they are. Apples grow on trees, carrots grow underground. A's bond with T's, or U's in the case of RNA, and G's bond with C's. Okay, so it looks like a ladder. Okay, here's what the nucleotides coming together look like, and then it looks like a twisted ladder, just like that. Okay, the two sides are complementary, meaning if you have A, T, G on one side, you have the complementary. You have, I forgot what I said, T, A, C on the other side. There's other types of RNA that are important. Messenger RNA is important in transcription. Transfer RNA is important in translation. Ribosomal RNA is important for the ribosomes and for translation as well. Okay, this one went a little long, I'm sorry. At this point, take a look at page 88 and go through that review, as well as reviewing the whole chapter. Come up with some post-it note questions for me, and we will talk about it during our Friday discussion, which is one of my favorite days of the week.